speaker, Claudio Villalobos Dintrans, who told me that he was originally from Chile and got interested in neuroscience through work from the Montemar laboratory in the 60s and 70s. And some of you might know that they also recorded in the local version, as Claudio called it, of the giant squid, and did some cool electrophysiology there. And today, Claudio will talk about optogenetic activation of the inhibitory nigro-collicular pathway and how it evokes paradoxical contralateral orienting movements in mice. So please, Claudio, take it away. You have to unmute yourself.
Okay. Is that okay?
Perfect.
All right, cool. Thanks for the invitation and running flawless and Neuromatch 3.0. Yeah, so the work that we've been carrying out involves the regulation from the basic ganglia and the inputs into the superior colliculus and how this controls especially orienting movement. So in the superior colliculus, and I'm going to show a picture of Villalobos because we are in South California. I know this seems warm, the rest of the country is getting cold, but we need to, the superior colliculus integrate a lot of sensory information in order to try to decide what's the best to transform those sensory inputs into motor actions for reaching different goals. In the case of volleyball, you need to see the speed of the wing, you need to look at the visual inputs of the ball to try to get the ball and get the point. When these motor actions, they get somehow intervene or you don't get the right motor action, you get, undecided effects that they can be paid for under certain circumstances. So the normal, the current model of how the superior colliculus and the basal ganglia interact to produce oriented movements is derived from the work from picosecond works from the 80s, where they see that during the eye movement, there's a reduction in the activity of the substantia nigra right at the time that the eye start moving. This led to create the hypothesis that to be able to move, to control eye movement, there's this inhibition of the substantia nigra into the superior colliculus that release the concept inhibition, allowing the cell for the superior colliculus to burst and produce and activate it to produce the contralateral movement that generates the eye movement. All this work has been done mostly in monkeys, and it generates this model where the substantia nigra plays a passive role, where the lower activity of this inhibitory inputs into the superior colliculus allows cortical excitatory inputs to produce these orienting movements. And this has been the textbook model of how the orienting movements have been developed. And it hasn't been challenged in the past 20, 30 years. However, a recent studies have suggested that the model might not be completely correct or not as simple as the original idea suggests. For example, in birds, which has an homologue of the superior colliculus in mammals, you see the connection between the area 8 and the BLM that is similar to the connections from basal ganglia and thalamus. When the activation of the area X, that is an inhibitory synapse through the BLM, you can see that there's an inhibitory, this is an inhibitory response, but after the cessation of the inhibitory response, there was some spike on the cells from the BLM that it started to create this idea that the role of these inhibitory inputs can actually not just be passive, but it could be actually active. And in results from our lab in monkeys, where they stimulate in the substantia nigra in the record cells in the superior colliculus, they see that in these panels, you see when there's an activation of the substantia nigra, you can see the inhibition of the activity of the cells in the superior colliculus, but there's a ramping of the activity. So it's not on associated with a more concentrated eye movement. So this association between the inhibitory inputs of the superior colliculus, the basal ganglia into the superior colliculus might produce a different model than the ones that we know from the original cells in monkeys. So my goal was to actually test this idea, and we did a simple experiment where we inject an opsin into this substantia nigra and activated the terminals of the nigro-collicular pathway into the superior colliculus in freely moving animals and see what happened, what kind of movement will we get, and whether we'll move contralateral or ipsilateral to the side of the stimulation. So the possible outcomes is that if we activated the cells from the nigro-collicular terminals, so not only stopping the inhibition, but increasing the inhibition, one of the outcomes is simply the superior colliculus gets inhibited, as the model suggests, and we'll have no movement. An alternative hypothesis is that the activation of the nigro-collicular pathway will inhibit the ipsilateral superior colliculus, allowing the increased activity of the contralateral superior colliculus. And because they project contralateral to the brainstem, they will generate ipsilateral movement. So these are all hypotheses that agree with the current model, and this is kind of what we expected. But what is what we see? So, and this is just videos of the mice, and you can see when we activate, actually the optoprobe is on the right side, so you can see when we put the light pulses, the mouse actually not only doesn't move or ipsilateral, but it has a strongly contralateral oriented movement that is evoked by the activation of the nigro-collicular terminal. And you can see in a mouse that it is being transfected into substantia nigra only with GFP. Activation of the light pulses doesn't produce these oriented movements. And this is just a quantification. We did like 4 or 5 mice, and all the mice, they show increased contralateral movements compared to ipsilateral movements, and there is significant movement compared to the control mice. So this is definitely an unexpected result that doesn't agree much with the previous model of how the basal ganglia and the superior colliculus interact to produce oriented movements. So we next wanted to see, so what's the mechanism that underlies this oriented movement that is evoked by inhibitory inputs. So we decided to test the outputs, the activity of the output cells of the superior colliculus, that the ones that they go contralateral into the brainstem. And we saw that the cells actually, with the inhibitory inputs, they do spike after the presence of inhibitory inputs, but the big majority of these cells. So these cells can spike with inhibitory inputs. So then we want to see, so what is the mechanism that it produces spiking through inhibition in the cells that they can trigger these oriented movements? So we decide to label these cells using a retrograde virus and using the same type of injections that we use for the in vivo experiment, and see using patch clamp on these specific cells whether the nigro-collicular activation can actually reduce this inhibition and spiking. And we can see exactly what happened is that's what we thought, that these cells from the alpha cells from the superior colliculus, when activated through the nigro-collicular pathway using optogenetics, they do present inhibition, and upon the cessation of this activation, the cells start spiking. So these type of cells, and you can see that there's no spiking, and when you do the optogenetic activation, the cells start spiking. So these specific cells, the alpha cells of superior colliculus, they do spike with increase of activity of the nigro-collicular pathway. And then we further want to see just a pharmacological corroboration that how they do it, and they do it through activation of the T-type calcium channels, which is the ones that are producing the rebound delateralization that you see at the end of the inhibition, and it produces the spiking of this cell. So the conclusions is that so we have the optogenetic activation of the nigro-collicular pathway evolves instead of just preventing movement. It involves contralateral movement in orienting movement to mice, and this orienting movement is produced by a rebound polarization and spiking in these cells in the outer cells of the superior colliculus. And this spike, rebound polarization and spiking is produced by a theta calcium channel that is producing these rebound polarizations and spiking in the cell. And at the end, I just wanted to acknowledge Michelle, and all the person in the laboratory, my collaborators, and our funding institutions. Thank you.
Awesome. Thank you so much, Claudio. We have a question in the Q&A by Marcus. Marcus asks, if excitation of the SNR causes superior colliculus excitation, what is the difference with the canonical model of disinhibition, as inhibition or excitation of the SNR would appear to have the same effect?
Right. So the canonical model says that the cessation of the activity of the substantia nigra, what it produces, it produces a disinhibition of the cells on the superior colliculus, allowing excitatory cortical inputs to excite the cell. What we're suggesting is that the cortical cells, the activation of the nigro-collicular pathway by itself can produce this orienting movement. So the role of the basal ganglia is not just passive, like a gate, but it's actually inducing these orienting movements by activating the output cells of the collicular through rebound polarization.
So can I follow up on that? So is that just because of the way you manipulate the cell's activity essentially through optogenetics in your experiment? Or do you think that that would also hold in like natural behavior?
Right. So this is another hole that there is in the knowledge of these systems, where it's commonly known that the cells from the substantia nigra spike between 20 and 40 hertz. But recent studies have suggested that there's an increase on the activity of the cells up to 100 to 100 hertz, which is physiologically correlates with the type of simulations that we have done. And that increase in activity in the substantia nigra might be correlated with behavioral. So we think that the substantia nigra also modulates its activity, and it not only decrease, but also can increase its activity, and this increase in activity could produce these orienting movements in the superior colliculus.
I have another question. So when you showed us the video of the mice moving with the stimulation, it seemed like that the movement of the mouse was continuous, despite the fact that the stimulation went on and off. And I would have expected that the movement was like, there was a movement, then it would stop. There was a movement again, it would stop. Can you comment on that?
Yes, that's exactly what happens. I'm sorry to mention the video it's speed up for time's sake. But that's exactly what happens. If you see a slow down video, you can see that not only the movement is not continuous, but the movement happens right at the end of the stimulation. So it's not like we're stimulating something, but you stimulate, and at the end of stimulation, the mouse moves. You stimulate again, and at the end of stimulation, the mouse moves again. In the preprint, or whoever wants to know more, I can send you a slow down video where you can really see this effect, and that's exactly how it happens. It's not a continuous movement, but it's a movement that happens right at the end of the simulation.
Yeah, so that was just the way that the video was a little too fast to notice that. Okay, cool. Yep. Thank you so much. Are there any other questions? Maybe someone from the panels? Any of the panelists have any questions? If not, then we can move on. Thank you so much again, Claudio. That was a great presentation. So next up, we have Priscilla Ambrosi, who is